The field of epigenetics, whose definition is highly contested, broadly refers to the study of heritable differences in gene function that cannot be explained by DNA sequence changes. The term epigenetics was first introduced by Conrad Waddington in the 1950s to explain how diverse cell types in the body could arise from one set of genetic material. Researchers have identified many processes thought to have an epigenetic basis, but there is still significant debate about many fundamental principles of the field. In this video, we will highlight important discoveries in epigenetics, key questions being debated by epigeneticists, common tools used to answer these questions, and finally, some current research in the field. First, let's review several key moments in the history of epigenetics. In the 1930s, Hermann J. Muller observed a phenomenon known as position effect variegation in Drosophila. He found mutant flies with mottled eyes and linked this phenotype to the variable spread of condensed heterochromatin that silenced the gene responsible for eye color. This would be the first identified epigenetic phenomenon where a phenotypic change was observed without a corresponding change to the genetic sequence. In 1959, Susumu Ono observed in female rat liver cells that one of the two X-chromosomes was condensed. 2 years later, Mary Lyon hypothesized that this condensed X-chromosome is genetically inactivated, that the choice of which X-chromosome to be inactivated is random, and that this inactivation is stably inherited by the cell's offspring. This process, now called X-chromosome inactivation, or XCI, causes females to be biological mosaics. In 1964, Alfred Mirsky published the earliest work on the role of histone modifications in gene regulation. Histones form the core of nucleosomes, which are the basic repeating unit of chromatin in eukaryotic cells. Mirsky studied how methylation and acetylation of histones affected RNA synthesis, and it is now known that numerous modifications alter the activity state of nearby chromosomal regions. In 1975, Robin Holliday and his student John Pugh, and independently Arthur Riggs, proposed that methylation of CpG dinucleotides in DNA might be involved in stable epigenetic silencing, for example during XCI. Adrian Bird and colleagues lent further credence to this idea in 1985 by identifying clusters of unmethylated CpG sites throughout the genome that were later associated with transcriptionally active promoters. He would later also discover regulatory proteins that bind methylated DNA, eventually repressing transcription. In 1984, Davor Solter, Azim Surani, and others observed that mouse embryos containing only maternal or paternal genetic material created via nuclear transplantation experiments did not develop normally. This marked the discovery of genomic imprinting, or parent-of-origin-specific gene expression. The first imprinted genes were discovered in 1991, where only the copy inherited from either the father or mother is ever expressed. One of these genes, H19, turns out to be rather unusual. Its final product is a 2.3 kilobase RNA that does not get translated into proteins. More of these long non-coding RNAs, or link RNAs, were soon discovered, including Exist, which is required for shutting down the X chromosome during XCI. Current evidence suggests that these RNAs may function as scaffolds to recruit regulatory factors. Today, researchers continue to work out how interactions among link RNAs, DNA methylation, and histone modifications regulate epigenetic processes. Now, let's turn to some questions being asked by epigeneticists. At the most basic level, scientists are still actively studying the mechanisms by which epigenetic marks, such as histone modifications and DNA methylation, are created, removed, and interpreted. Researchers continue to characterize the enzymes that carry out these functions, as well as how the marks interact with the transcription machinery to activate or repress gene expression. A deeper question that arises is whether there exists an epigenetic code analogous to the well-defined genetic code that dictates how information in DNA is translated into protein sequence. Researchers are trying to determine if the combination of epigenetic marks form a similarly predictive code that will one day make it possible to deduce the expression pattern of every gene. Recently, scientists have been interested in the biological roles of lincRNAs. While a prevailing model is that lincRNAs help recruit epigenetic factors to specific genomic locations, their exact mechanisms and whether all lincRNAs function similarly are still being studied. Finally, because epigenetic marks are chemical add-ons that are not simply replicated along with DNA, scientists are still trying to learn how the marks continue through cellular generations. Even more controversial is the potential transgenerational inheritance of certain epigenetic processes. Because it is observed that epigenetic marks are dramatically erased or reprogrammed early in embryogenesis and again during gamete formation, how and whether these transgenerational phenomena actually occur remains hotly debated. Let's now look at some tools being used in the study of epigenetics. DNA methylation is most commonly detected by bisulfite analysis, a process to change unmethylated cytosine residues to uracil, which are then detected as thymine in sequencing reactions. Comparing sequences before and after bisulfite treatment allows researchers to identify the locations of methylated DNA. Another method to assay DNA methylation status is to digest DNA with methylation-sensitive restriction enzymes, which can only cut unmethylated DNA. Immunoprecipitation, or pull-down techniques, are used to identify DNA or RNA sequences associated with specific features. Chromatin immunoprecipitation, or CHIP, isolates DNA bound by particular protein factors or histone modifications, whose sequence information can then be analyzed by PCR or sequencing. On the other hand, methylated DNA immunoprecipitation, or MEDIP, is used for isolating and enriching methylated DNA. RNA immunoprecipitation, or RIP, and chromatin isolation by RNA purification, or CHIRP, can respectively determine the protein partners of a non-coding RNA, or its genomic binding locations. After reviewing the tools of epigenetics research, let's look at some of their applications. 5-hydroxymethylcytosine, or 5-HMC, a recently discovered alternate form of cytosine methylation, has garnered much scientific interest. In this experiment, researchers performed a chemical reaction that specifically adds a glucose molecule to hydroxymethylated DNA, and then treated the DNA with restriction enzymes that either only cut unmethylated DNA or cut all methylated DNA except when modified to be glucosylated 5-HMC. This analysis, followed by PCR, allowed discrimination between types of methylation at specific DNA bases. To understand the role of lincRNAs in epigenetics, researchers can perform CHIRP to identify their genomic binding sites. Here, DNA-RNA protein complexes in cells were first tightly cross-linked with chemicals. Chemically tagged antisense oligonucleotides were then allowed to bind to the lincRNA, and proteins with high affinity for the tags were used to pull down the complexes. DNA sequences that interact with the lincRNA were then identified by sequencing. Finally, epigenetic processes such as X-chromosome inactivation can be studied at the single-cell level using microscopy-based techniques. In this experiment, fluorescence in situ hybridization for exist RNA was combined with immunofluorescence against known histone modifications. The lincRNA and histone marks could then be co-localized to reveal possible functional relationships. You've just watched JoVE's Overview of Epigenetics. In this video, we looked at the history of the field of epigenetics, some of the prominent questions, and tools of the field, and specific examples of epigenetic research. As always, thanks for watching.